Hello, my name is Hidmark, and this is the second part of my Innate Immunity lecture series. It started with the first part, mucosal defenses. I am now doing a part which is called Innate Immunity, cells, receptors, and how bacteria fight them. I will divide this lecture up in pieces of around 10 to 15 minutes and put them into a playlist. This is one of the lectures that I give at the University of Heidelberg to the master students of the infectious disease program.
So the immune system is generally divided up into types of immunity, the innate immunity and the adaptive immunity. The innate immunity is the very fast immunity which does not require that the pathogen has been seen before. Innate immunity serves to prevent the initial spread of the pathogens, also to make cells less infectable and to eat up and remove pathogenic material. The adaptive immunity is later on stimulated by the innate immunity and takes over after a few days. The definition of the innate immunity is the quick response time, that it can recognize self from foreign using the receptors we will talk about, the antimicrobial activity, mainly of the macrophages, and that is actually localized through the whole body. As you see on the time scale on the picture on the right, the reaction times of the innate immunity is counted in hours. What we will talk about here is the phagocytes and mainly the macrophages, which are depicted in green, and the dendritic cells, which are pink. We will not talk a lot about mast cells, while the importance for infection is not clear. We will also come back to the ILCs, and NK cell is a type of an ILC, possibly. And we will also talk, finally, about complement. So if we look at the response times, the innate immune response is only really activated during the first part of the infection. The adaptive immune response takes over after about 5 days and will eradicate the infection. So the purpose of the innate immune response is to limit the burden of the pathogens. One reason why the innate immune response is only active in the beginning of the infection is that there are mechanisms to turn off the innate immune response after an initial response. The fact that the innate immune response has been downregulated in the beginning of an infection may give the opportunity for secondary infections later on. This is a picture that was shown yesterday, and I will quickly go through it again to recap. On the very left side, you have the constitutive innate immunity. Here you see the barrier of the epithelial cells, and once that is breached on the inside, granulocytes like neutrophils will immediately attack bacteria. Macrophages will take up bacteria, and so will dendritic cells. Both macrophages and dendritic cells will present the bacterial components or viral components to adaptive immunity. The dendritic cell can prime CD4 and CD8 T cells, and the B lymphocytes are requiring CD4 help. Once the B lymphocyte is producing antibodies, the complement can be activated by the antibodies. So the complement is thus both dependent on the adaptive immune response, but it can also function independently of the adaptive immune responses, and that's why we're going to talk about it here. In the very center of this picture is the monocyte or macrophage, which is the heavy worker in the innate immune system. The monocyte, the macrophages, and the dendritic cells, which is called the myeloid type of cells, are expert at recognizing pathogens and signal to other cells. There are several different pathogen recognition receptors in the innate immune system. The extracellular receptors can be divided up into 2 types. It is the TLRs and the lectins. The TLRs will, on the surface receptor TLRs, will recognize bacterial cell wall components. The lectins, for example dectin 1, will recognize a component of the wall of different yeasts called zymosan. In the cytoplasm, there are 3 different types of receptors. The NLR, which we will come back to, recognizes bacterial peptidoglycan. The RLR, the Rig-like receptors, recognizes viral RNA. And the cytosolic DNA sensors will recognize microbial DNA in the cytoplasm. There are also several TLR receptors in the endosome, which will recognize taken up pathogens, which is very common in the phagocytic cells. These TLRs will recognize mainly nucleic acids. The TLRs are a conserved type of receptor expressed on many different tissues. These receptors can recognize a wider range of patterns released either from dead cells or from pathogens. The Toll receptor was initially discovered in Drosophila, where it mediates the dorsal ventral polarization. But also in the fruit flies, the Toll receptor is an innate immune receptor. Homologs of the Toll receptor are called the Toll-like receptors, and they are found in all mammalian cells. The Toll-like receptor, the TLR, it consists of one single membrane-spanning region. The extracellular region is characterized by the leucine-rich repeats, which are flanked by characteristic cysteine-rich motifs. In the cytoplasmic part of the Toll-like receptors, there is a motif called TIR, which is a Toll IL-1 receptor domain. The TIR domain is also found in the receptors for cytokines IL-1 and IL-18. The specificity of the TLR system is further enhanced by the ability of TLR2 to heterodimerize. For example, dimers of TLR2 and TLR6 are required for the response to peptidoglycans. The specificities of the TLRs can also be influenced by various non-TLR accessory proteins. This is best defined in the TLR4 response to LPS. LPS first bind to the soluble LPS binding protein in the blood or extracellular fluid, and this complex serves to facilitate the delivery of LPS to the surface of the responding cell. An extracellular protein called MD2, myeloid differentiation protein 2, binds to the lipid A component of the LPS and forms a complex that interacts with TLR4 and initiates signaling. Here is a table over the different types of ligands and synthetic ligands which exist for the different TLRs. This table can be used as a reference table, but to some points here, TLR3 is recognizing viral RNA and the double-stranded motifs in the viral RNA during replication. This receptor can also be triggered by poly-IC, which is a synthetic molecule that does not exist in nature. The poly-IC consists of cytidine and another complementary strand of inosine. Inosine is a nucleotide which is normally not found in the cell. The TLR9 is recognizing DNA from bacteria and virus, and in the nuclei the DNA is methylated. However, DNA which is produced by bacteria or in the cytoplasm is not methylated. Also, the TLR9 is strongly activated by certain sequence events in the DNA, which is common in the bacteria but not very common in the human. TLR7 and TLR8 are tandem repeats on the X chromosome. They were first described to recognize the antiviral component imiquimod, but only later on has it been shown that the TLR7 and 8 will recognize single-strand RNA, and the basis for recognition here is uridine content, and also there is a sequence specificity. TLR4 has been described to recognize very many different ligands, and part of this broadness has been due to LPS contamination in the preparations used. However, TLR4 does seem to be a very promiscuous receptor, and it has also been described to recognize crystals of urea, which is released from necrotic cells. The most common molecule for downstream signaling of the TLRs is the MYD88 molecule. The MYD88 adapter molecule is being used by all the TLRs except for TLR3. Downstream signaling pathway of MYD88 I'm going to talk about in the next slide, but the consequence of the MYD88 activation is the production of the cytokines, so TNF and IL-1 and IL-6, the chemokines, endothelial adhesion molecules, for example E-selectins, and also upregulation of co-stimulatory molecules. Activation of MYD88 leads to an acute inflammation. The TLRs inside of the endosome also activate MYD88, but they need some accessory proteins in order to do that, for example UNC93B. The TLR4 activates both MYD88 and TRIF, which is the only adapter protein of the TLR3 pathway. The TRIF will in turn activate the interferon regulatory factor and cause production of type 1 interference. So TLR4 is a good example of the TLR signaling pathways, since TLR4 will signal through both the same pathways as all other TLRs and also via the TRAM-TRIF pathway that TLR3 uses. So the standard MYD88 signaling is going via IRAC towards TRAF6, and there are many different other adapter molecules in this pathway which have not been included here because it's a lot of names and it looks very complex. But what TRAF6 does is that it releases NF-kappa B from the inhibitory kappa Bs, and that allows the NF-kappa B to go into the nucleus. It also activates the MAP kinases. The endosomal signaling from TLR4 is mainly activating the TRIF pathway. TRIF is another adapter molecule which in turn is activating TBK1. TBK1, or tank binding kinase 1, is also downstream of the antiviral response and the cytoplasmic receptors for RNA. TBK1 will phosphorylate IRF3, which is essential for induction of type 1 interferons, which we will talk a little bit more about soon.